Okay, hello everyone, and welcome again to a new webinar as part of the Innovation and Precision Eye Health webinar series. I'm Ray Najjar. I'm an assistant professor in the Department of Ophthalmology at the National University of Singapore, and I'm a visual neuroscientist. And today it gives me great pleasure to welcome our speaker, Professor Manuel Spitschan, who is appointed to the Rudolf Musbauer tenure-track assistant professorship at the Chronobiology and Health at the Technical University of Munich. Manuel is also a group leader at the Max Planck Institute for Biological Cybernetics in Germany. So Manuel is a very close collaborator and friend. We actually have similar tracks almost. He's also a visual neuroscientist. And he basically studied psychology at the University of St. Andrews and received his PhD from the University of Pennsylvania. From 2016 to 2017, he was a postdoctoral fellow at Stanford University. And from 2017 to 2021, he was also a Sir Henry Wellcome Fellow and was promoted to university research lecturer before taking on the position that he currently holds at the Technical University of Munich and Max Planck Institute. So Manuel's interest was and always is about light and how light beyond vision can impact health. And he also focused on the impact of light specifically on human circadian and neuroendocrine physiology through actually the pathways, and studied the pathways connecting the eye, the retina, to the brain, to the hypothalamus of the brain. So without further ado, actually, I want to welcome you, Manuel, to give this talk. Let me just briefly mention a couple of pointers to our participants. You are all on mute, that's normal, but if you have any questions, please feel free to ask these questions in the Q&A section at the bottom. We have 20 minutes at the end to answer your questions and have a discussion with Manuel, but also another panelist member, Dr. Daniel Tay from NUS Ophthalmology. And please stay until the end. We will have a very exciting discussion at the end. So without further ado, Manuel, can you please tell us, what does the eye tell the brain? All yours.
Excellent. Thank you for the very, very kind introduction, Ray, and also for the invitation to deliver this seminar. I think it's also great to see so many people joining here. And I promise you, I think there's going to be a lot of content that will be interesting to you and will be exciting. My name is Manuel Spitschan, and delighted to talk about how or what the eye tells the clock today. And before I get started, one of the things that I kind of want to highlight is that we all have some relationship to light, right? I think right now you're probably sitting in an office or somewhere else where you're exposed to different light sources. Some of them are probably suboptimal and some of them might be too bright. And what I'd like to get, what I'd like to start with really is a little bit of an understanding of the audience's knowledge in the topic that I'll be talking about. I understand some of you are eye professionals, optometrists, ophthalmologists, but also from different disciplines. And so we prepared a couple of questions that we'd just like to ask you just at the beginning of the talk to get a little bit of a sense of your background knowledge. And so there should be a Zoom quiz popping up. There's various questions, 3 questions that I want to ask you. And so everyone, please do answer these questions. I'll give you maybe half a minute and then we can move forward with the talk. This is really, it's not a quiz, right? This is not, even though it says quiz, it's not a quiz in the sense that you will be examined on it, but it really just helps us understand a little bit, you know, what is your background, where you're coming from, what's your baseline knowledge. If you are unsure about things, don't worry, we'll come back to these questions throughout the talk. So I think we can leave the questions up while I move on probably with the talk. At some point, we should probably close them. So light has a very, very important influence on human health and well-being, and it's the light detected through the eyes that does this. So the focus of today's talk will be ocular light exposure and its effects. And we know there's a couple of ones that are very, very important to our health and well-being. Here's a couple of ones that are listed. We have influences of light exposure on the production of hormones, core body temperature. Light can modulate our alertness. This might be specifically the case if you are having a post-lunch dip. Maybe light gives you a little bit of a boost in the afternoon. The timing of our circadian clocks, of our biological clocks, sleep, mood, and also, and I think this is very relevant to this audience, eye health with specific interest in myopia. And so you can see how light is not just something that helps us see and navigate in the world, it is also something that we need to optimize and control to optimally deliver good lighting scenarios in the world, in the real world. And of course, there are mechanisms that underlie these various effects that I'll want to talk about now. So I think we've all seen pictures like this where we have eyes, light entering the eyes. And yeah, if you look at the visual system, the standard textbook understanding is, of course, that we have various pathways that connect the eyes to the visual system. The primary visual cortex after being relayed by the lateral geniculate nucleus, and then higher order visual areas, V2, V3, HV4, and MT. And so these are really, if you open up a neuroscience textbook and you look at what is the visual system, you'll find something like that, basically projections of the retina to the brain to visual cortex. And of course, these functions or these areas serve various important functions, seeing color, motion, detail, and helping us navigate. Around, I'd say 40 years ago, there was knowledge developed and discovered that there's another pathway that connects really the eyes with another part of the brain, and that is the suprachiasmatic nucleus. And so there's the so-called retino-hypothalamic tract that connects the retina with the hypothalamus. Now, if neuroanatomy is a little bit something that you might have studied a few decades ago, the hypothalamus is incredibly important for vegetative functions. And one of the functions that the hypothalamus houses, in a way, is the control of the circadian clock. And so we have a direct connection between the eyes and our circadian clock. The SCN, that's the suprachiasmatic nucleus, projects a number of different targets. And one of the things that it subserves as is the neural control of the production of melatonin. So there's neurons in the SCN that fire depending on the circadian time, but also light has an influence that acutely affects the suppression of melatonin in the pineal gland. We now know, of course, this is knowledge that we've had for about 20 to 25 years. There's special cells in the retina that underlie this pathway, and I'll come back to them in a second. It was also in the question that we just asked you. And it's really fascinating to see kind of these two parallel pathways, right? The pathway to go to primary visual cortex and the pathway to go to centers that control what we generally consider non-visual functions. Non-visual functions is a little bit of an umbrella term for all the things that are light-mediated but don't contribute to vision. The circadian clocks, of course, are very, very important. Almost every cell in the body has a circadian clock, and many organs follow circadian rhythms, right? And so the control of the SCN as the also sometimes referred to as the conductor of the circadian clock is really important to keep the peripheral clocks in our bodies synchronized. Now, light exposure is really, really important. And so just before I dive a little bit more into the mechanisms, I just want to highlight a paper that I was a co-author on that was published last year. This is recommendations for daytime, evening, and nighttime indoor light exposure to best support physiology, sleep, and wakefulness in healthy adults. And so what this paper did is recommended or produced recommendations, basically recommended light levels for the daytime, the pre-sleep, and the sleep environment based on a really thorough meta-analysis of different types of data. And so this is what we, of course, call a dose-response curve. Here, I'll just show you in panel A, melatonin suppression. The brighter the light gets, here expressed as melanopic equivalent daylight illuminance, the more melatonin suppression you get, right? So the brighter the light, the more melatonin production will be suppressed. You can see similar effects in the entrainment of the circadian clock, that's shown in panel B, and then also similar dose-response curve when it comes to the control of alertness. And so when you put all these curves together, you end up really with a summary dose-response curve. This is across many different studies based on which then we derive these recommendations. One of the key things here, though, is if you look at melanopic EDI, that has a unit in lux. And so you might have come across lux in your everyday life. If you don't have a background in lighting design, you might nonetheless know lux levels, for example. You might know that light and illuminances are expressed as lux. And what we have here with the melanopic equivalent daylight illuminance is a unit to express the light levels with respect to circadian physiology. And so melanopic here is the operative word. It contains the photopigment melanopsin, and that is the photopigment in the eye that helps us reset our circadian rhythms, suppress melatonin, and also modulate alertness. And so just as a way back, you also know this picture very well. Light enters the eye through the lens and it gets imaged in the back of the eye in the retina, fine layer of cells. And until about 25 years ago, the textbook knowledge was there's cones and the rods. The cones, of course, allow us to see in daytime lighting conditions, color, motion, spatial detail, while the rods help us see under dim conditions, but with very, very low visual acuity. And in 1998, in the late 90s, another cell was discovered in the retina that was light sensitive, and these are the so-called intrinsically photosensitive retinal ganglion cells, also abbreviated as IPRGCs. This is a special set of retinal ganglion cells that are light sensitive without input from the rods and cones. So they express a photopigment in their membrane and in their processes that renders these cells directly light sensitive. So under some manipulations, molecular manipulations, when you knock out the rod and cone input, these IPRGCs are still able to signal light intensity. And that is because they express an opsin that is called melanopsin. This is why we had the melanopic light exposure. And importantly, melanopsin is very different in terms of the wavelengths it prefers compared to the rods and the cones. So this is really fundamental biology, but it also, over the past 25 years, has led to serious implications for how we think about how we should light our rooms, our spaces, our buildings, because, of course, if the spectral sensitivity of these melanopsin-containing cells is different, we need to incorporate this in our understanding of how we should use light in the built environment. This is the way that I've already shown you just now in a different view. Light hits the eye, enters the eye through the lens, hits the retina, and of course triggers a cascade of different responses. And of course, the primary or the visual system primarily then is illuminated, as it were, through the LGN cortex pathway, where functions are supported, including visual processing of color, space, and motion, as well as higher level visual processes, including visual working memory, the perception scenes, and any other higher level visual processes, right? What we typically consider vision or visual perception. And then, of course, we have another pathway, and that is through the SCN, the retinal hypothalamic pathway that underlies the non-visual effects of light, the melatonin suppression, circadian phase shifting, and the modulation of alertness. So two rather different pathways, very different functions, but of course, everything goes through the retina, right? And so one of the things that I've tried in my career is really bring together our understanding of the clock, our understanding of the effects of light on human physiology, and behavior, back to the retina, right? Back to the fundamental mechanisms that process light at the first stage, at the front end. And one of the questions that I've tackled in there is, how do we measure the contributions of a given photoreceptor class to these outputs, to these functions? So light enters the eye, hits all photoreceptors in some way, and therefore produces responses in all of them. Now, how can we separate out what a given photoreceptor class is doing? And so I want to describe a method that we've used to study the photoreceptor contributions to pupillary light responses and also to melatonin suppression. And in a visual neuroscience sense, I think this is a very powerful technique because it allows us to dissect how different photoreceptors contribute to different processes in the living human eye. Now, if I was working in animal studies, I'd have some type of genetic method available. Here's schematically shown a mouse retina. And one method that might be available, or one class of methods that might be available, would be one that looks at functions supported when the cones and rods are knocked out, right? So a question being asked is, which function is preserved in the absence of the cones and rods? You can ask the inverse as well. You can ask the question, which function is this deficient in the absence of the melanopsin-containing IPRGCs? And so with these two broad techniques, you can then put a picture together of what are the necessary and sufficient contributions of these photoreceptor classes. And of course, in humans, these techniques are not available, so we have to be a little bit more inventive, a little bit more exploitative of physiology to really get at what a given photoreceptor class is doing. And so generally, these techniques, of course, are not available, and there are some genetic cases or one specific group of participants that I want to come back to in one study that we ran a few years ago, looking at what happens when you don't have functional cones. It's a small-scale study, but nonetheless opens up a way, a framework of thinking about the photoreceptor contributions. Now, I mentioned we need to exploit physiology, and one of the things that is different between the photoreceptors are that they have different spectral sensitivities. These are the wavelength preferences of the photoreceptors, the S cones, the M cones, and the L cones, melanopsin, and the rods. And what we, of course, realize that because the photoreceptors have overlapping spectral sensitivities, if you use a single wavelength light, such as those generated by an LED or a laser, one challenge is that these lights will not uniquely activate a single photopigment, but instead they give you a broad range of activations across all photoreceptors. Now, you can play a little bit of a trick where you select two different wavelengths and then compare them, which I think is a very, very nice method. But generally, you still activate all photoreceptors in this technique. So one of the basic photobiological principles that we can exploit is what's called the principle of univariance. Here I'm showing you the spectral sensitivity of one photoreceptor, and I'm showing you three hypothetical lights. Let's call them E1, E2, and E3. The principle of univariance states that the photoreceptor doesn't know whether the light that it is seeing has a different wavelength or a different intensity, because the only thing it does, it captures photons according to its wavelength sensitivity, its spectral sensitivity, and converts it into a scalar output, the photocurrent, or then later on spiking. And so what you can then basically say is you find three different lights that will produce the same output in the photoreceptors. If you do the math, 0.5 of the spectral sensitivity times 1 is 0.5. 0.5 times 1 is still 0.5, and again, this is on the other flank of the photoreceptor. And so if you integrate the three lights, E1, E2, E3, will have the same response, of course, in the photoreceptor. We can exploit this. What we can do is we can select two wavelengths, which if we alternate them, we keep the excitation of one photoreceptor constant. And so in this case, I found two lights that produce the same response in my S cones while producing a difference in response, a differential response in melanopsin. So now we have something that we can use to, of course, this needs to be scaled up to more photoreceptors, but we now have a technique in hand based on a fundamental photobiological principle with which we can modulate the activation of different photoreceptors. We call this method the method of silent substitution, and I want to show you a couple of studies that we ran using the method of silent substitution, where we looked at how do different photoreceptors contribute to different functions. We need to, of course, when you have 5 photoreceptors, do a little bit more spectral engineering, but what we can do is we manipulate light spectra using multi-LED chips, for example, to expose specific mechanisms. I want to highlight one study in which we examined how different photoreceptors contribute to the pupillary light reflex. We had a light source in which we were able to control the light, the spectral power distribution of the light, stimulating a pharmacologically dilated eye while tracking pupil size in the other with an IR camera. And so we did this as follows. We measured the pupil response to different photopigments at a given temporal frequency, recorded the pupil responses over many trials, took an average across trials, and then fit a sinusoid at the modulation frequency. This is just signal processing, but it helps us do two things. First of all, we can estimate the amplitude of the pupil response and the phase, right? So what is the temporal relationship between the pupil response and the light the eye is seeing? This is a very dense data plot, but basically what this shows us is across temporal frequencies, so very slow sinusoidal modulations at a scale of 0.01 hertz to 2 hertz, so twice a second. We stimulated the S cones, melanopsin, L plus M, the luminance channel, And what we can see is distinct pupil responses depending on which photoreceptors are activated. And I want to highlight one here, and that's the L&M cones at 0.05 hertz. This is seeing, so the stimulus phase here is you have an increase in contrast at the beginning and a decrease at the end. So with an increase in contrast, you have a constriction of the pupil, and with a decrease in contrast, you have a dilation of the pupil. And that's what you would predict given what you know about the pupil. Except if you look at the S cones, you see something else, and that is an anti-phase response. With increase in S cone contrast, the pupil actually dilates. So we can summarize this in a plot that looks like this, where we have the amplitude as the radial eccentricity and the angle as the phase. And we can look at how do these responses to LNM, melanopsin, S cone cluster. LNM are in the first quadrant, melanopsin here in quadrature phase, interestingly. And S cones, and this is the key finding from this study, we looked at what we would predict from physiological data, neurophysiological data. And it turns out that Dacey in 2005 found that S cones provide an inhibitory input into the melanopsin containing RGCs. And so we'd predict if they provided inhibitory, so anti-phase input, the S cone points might lie somewhere here. And because this would be in anti-phase and 180 degrees from the LNM signal. And indeed, on average, we find the S cones actually contribute to the pupil response in an opponent fashion. So increases in S cone activation lead to a dilation of the pupil. Decreases in S cone activation lead to a constriction of the pupil, which is flipped, right? Because typically what you have, you increase the light, pupil goes small, you decrease the light, pupil goes big. We find this at different temporal frequencies as well. And so this is really, to us, an interesting hallmark of how photoreceptor signals contribute to the IP-IGC activity, because the S cones have this inhibitory response or inhibitory input. We also see that the different photoreceptors have different temporal responses, with LNM having a bandpass response and S cones and melanopsin preferring slower frequencies. And this is entirely consistent with what we know about the light integration mechanisms in the circadian clock and the IP-IGCs. They prefer slower visual signals, mostly because, of course, their goal is to track changes in light intensity across time of day, and not like the cones to encode changes in scene content or, the arrival of a predator or some other thing. So we asked, I think, then, what is this role of S cone opponency? Might it be a twilight detector? These are some fun data where we collected light exposure, nighttime spectra under conditions of no light pollution. And what happens, if you're a photographer, you probably know about the golden hour and the blue hour, but you see a distinct shift in the overall spectrum. Right? In the blue hour, the wavelength content of natural illumination is very much shifted to shorter wavelength. And it turns out if you have S cones that contribute to this processes in an opponent fashion, these might be ideally suited to pick up this specific signal. There's some work in mice now from Brown and colleagues that has also shown that there are these signals in circadian photoentrainment in mice. And in humans, we've just published a preprint in which we don't find this, but of course, this is very specific to the conditions. One of the things that we were also interested in then is the role of S cones in melatonin suppression. So we just looked at the pupil. Now let's look at something that's fundamentally non-visual, which is the suppression of melatonin by light. I already mentioned you, evening light and nighttime light suppresses the production of melatonin. And now the question is, through which photoreceptors does this happen? We know melanopsin plays an important role, but there is some data that shows that the S cones might also play a role. This is, of course, an area of active investigation. There's lots of work still trying to figure out how do different photoreceptors contribute to these processes. One of the things that Tim Brown showed in a meta-analysis was that there are some uncounted data for the S cones for very short wavelength lights. And so we put this question to the test. If you generate two lights, again, using the method of sound substitution, that differ in how much they activate the S cones, do you see a difference in melatonin suppression? So with clever spectral engineering, we built these spectra, these scenarios that differ in almost a factor of 100, so 2 log units, factor 83, in how much they activated the S cones. Of course, because the S cones contribute to color vision, these are very different lights, right? They look very different in color. You can't generate S-cone isolating stimuli that are identical. But what we examined was how do human observers respond in their melatonin suppression to these two stimuli. So this is a very specific protocol with 88 minutes of light exposure at a mean level of 170 lux. And we collected saliva samples every 30 minutes to ask, do these two scenarios that differ maximally in the amount of S-cone stimulation lead to a difference in melatonin suppression? The answer is we couldn't find an effect. And so with about 100-fold difference in S-cone activation, or 80-fold difference, we do not see a difference in melatonin suppression. We also don't see a difference in sleepiness or vigilant attention. And so what this would lead us to believe is most of the melatonin suppression effect is due to melanopsin, with the S-cones not playing a role. Now, if you think back about the S-cones driving pupil responses, you might also say, wait a minute, you did not control for pupil size, which of course we did not in this specific study. And so you might ask, well, there might be an effect from S-cones, but it's counterbalanced by the difference in pupil size. And so this is, of course, something that we would have to test in a specific paradigm. We did see, though, that there's differences in perceived color temperature. Overall, people did not like the light exposure that they were exposed to. And then overall, there's also, across the evening, a little bit of a decrease in general well-being. I think people are just happy to leave the lab. So you see this little uptick after, just before they are allowed to leave the evening protocol. And so with this data, we weren't able to find any S-cone effect, which I think is interesting. And I think also helps us then understand a little bit, how should we think about the role of cones in melatonin suppression. I mentioned at the beginning of my talk, we ran a study in a group of patients. And so the group of patients that we worked with, this is a very small scale study, was people with congenital achromatopsia. This is an eye condition in which a genetic mutation leads to dysfunction in cones, basically rendering these patients severely light avoidant, severely light sensitive. And so we worked with a geneticist who was able to genotype 6 congenital achromats. And we first examined, their light preference. And of course, you see light avoidance as a function of, approximate light intensity, almost always avoiding direct sunlight. And then, with lower light levels being a little bit more preferred. And so what we did here was a range of different things. We examined these discomfort ratings. We asked about filter uses. We did actigraphy and sleep logs. We did melatonin measurements, genetic confirmation, and then also statistical modeling. And I just want to highlight one piece of evidence that we collected here. We found normal rest activity cycles in these patients. So these folks are avoiding bright light exposure and typically use filters to stop down their light exposure. And also in the participants where we were able to get melatonin data, we found relatively normal melatonin phase angles. And so, of course, this helps us answer the question of how cone-mediated vision or lack of cone-mediated vision affects circadian rhythms, either pointing to the role of non-photic inputs into the circadian control, or some adaptation processes which recruits the rods and the melanopsin cells at different light levels. And so, of course, there's a lot more functional work that can follow from this. Now, I want to bring us back to the real world. These were all laboratory studies or relatively well-controlled studies. And when we are just living our daily lives, we're exposed to a range of different spectra. Here I'm showing you on the x-axis wavelength, on the y-axis radiance, or the amount of energy per photon, per wavelength. These are daylights that have relatively broad spectral power distributions, and we have artificial light. And of course, depending on what makes the light in the artificial light sources, the spectra will be different. With LED sources, they all come in different shapes. This is fluorescent lights. I still have some bulbs here or some tubes in my office. And the interesting thing here is, of course, you have 3 light sources that have the same or similar correlated color temperature, around 2,700, 3,000 Kelvin, the same lumen, but very different spectral power distributions. And this is one thing that, again, I want to bring home to you, which is the spectrum, right, is, of course, one factor that we need to consider. But these 3 spectra have the same, brightness appearance to us, but will have different effects on the melanopsin system. And so when we are looking at how do we assess the effects of light on human health and well-being, we can't just look at lux, we need to look at melanopic or melanopsin relevant quantities. We coined a term called the spectral diet, which basically encapsulates the light exposure that we are exposed to, and we've done some work looking at how could we capture this, how do we understand the totality of light exposure that people get throughout the day. This is a little bit of framework. You have light from all kinds of sources and scenes that is defined in time, space, and spectrum. We typically measure these with near corneal irradiance sensors, or wristwatches, or some things that you can pin, get personal light exposure patterns, and then summarize them. Of course, the real biological quantity that underlies most of the effects of light will be through retinal irradiance, which, of course, the only instrument that can measure retinal irradiance is the retina. Right? And so we have a little bit of a gap between what we can measure and what actually is the biological quantity that's driving it. These are some light sensors that have been developed in the past, little pins or watches or spectacle-worn devices. We work with a sensor as well that is clipped to glasses, and we've just been finalizing measurements and tests with a sensor that we're hoping to miniaturize in one of the next steps. This is a 18-channel sensor with UV exposure as well, and we're working on integrating this and making it a little bit smaller. One of the activities that we're also following here is we have a technical committee within the CIE, and what we're doing there is trying to standardize some of these methods for characterizing them. But ultimately, you need one of these sensors to capture personal light exposure. This is an example of a data set that we're just publishing, where we have 24-hour measurements with a corneal plane sensor. And first of all, you can see this is from, I think, 17 subjects. You see, of course, differences in their light exposure depending on what their day looked like. But you can also see that some people have a lot more varied light exposure across time of day than others. And of course, understanding these motifs, these statistical regularities in light exposure will be something that we'll be focusing on in the near future as well. Here's a summary plot of these. This is from all the participants. The light exposure, of course, as you enter the evening, you'll have less light exposure. And then we find, of course, a correlation between what we call the photopic illuminance and lux with the melanopsin quantity. This is one thing this tells us is, most of the time, melanopsin or melanopic EDI and photopic illuminance are correlated, right? So the world is relatively regular statistically when it comes to our light exposure. I want to come back to pupil measurements. We've done a relatively large-scale study where we looked at pupil responses across age. This was led by Raphael Lazar, who's a PhD student in Basel. And we built a system where we had a spectrometer and an eye tracker combined in one, where we were able to collect both pupil size and spectral measurements at the same time. Here's some examples of some of our individuals participating in this experiment. This is part of a calibration procedure where we ask people to basically give us as many snapshots of different angles of their eyes as possible. And what you can immediately appreciate here is, so I think some people find this incredibly creepy here, this type of video. I think it's quite fascinating, and particularly if you work with eyes, I think this is just something that just genuinely pops out to you. But what you can appreciate here is under the same nominal lighting conditions, you also have differences in pupil size, right? So there's individual variability in pupil size, and the factor that we looked at here is age. So what we examined was across different light intensities, how does age drive pupil size under real-world conditions? Here's two examples. This is, again, on the x-axis, melanopic EDI for an 18-year-old and melanopic EDI for an 87-year-old. These are the two youngest and oldest participants in the group of participants in our sample. And so there's two things you can see. First of all, you see the effect of light. Pupil size goes down with increasing light intensity under real-world conditions. I think you would expect this. This is textbook knowledge. You see this in both age groups or age examples here. But you also see senile meiosis, right? You see a constriction of the pupil, smaller pupils in the elder population. So this is just two examples. We can replicate these effects, of course, with a larger group. And this is very much in line with what we know from laboratory studies. You see a reduction, of course, of the slope of this light pupil response curve with age. Now, I want to come back to the light level recommendations that I started with. We have recommended light levels for daytime indoors, the pre-sleep and the sleep environment. One of the steps that I think should be following is, how do we carry these recommendations to policy? What can we do with them? We can write recommendations, but ultimately, can we turn this into legislation or regulations in some way so that people get the right light exposure at the right time? And that's, of course, something where we are very much working at the science policy interface, but nonetheless is important work in our views. One of the things that we've started to do is try to capture actual real-world variability of light exposure. And so this is both a very, very involved technical setup where we're collecting data with a spectrometer, with a colorimeter that collects data over time, and a hyperspectral camera, basically allowing us to characterize how light changes across different scenes, and then we could use these data to relate them to the recommendations, right? So an important question is, are people actually getting these light exposure levels, and what scenes or what types of scenes and categories are promoting reaching these levels, right? So if we say we need at least 250 melanopic EDI lux, 250 lux melanopic EDI during the day, but 90% of offices don't reach this, I think this gives us some cause for concern. And so we have a relatively involved protocol in which we're measuring lots of different things at the same time, and then through machine learning and big data techniques, try to poke out insights from these relatively large data sets. Another area in which we're moving to the real world a little bit is to understand behavior, right? So we can examine mechanisms in the laboratory. We can ask questions related to what photoreceptors contribute to what process. But one of the things that we don't do, if we do this, is understand what actually people do when they are in a room, right? When they say, I want to turn the light on, or I find this too dim. And so we developed an instrument that allows us to, in the first pass, in the first attempt at this, capture behaviors that support light exposure. And so as people engage in a variety of behaviors that modify light exposure, we need to understand what are these behaviors and how could we modify them, right? One aspect is to say, let's change the LED technology, let's change the light bulbs. Another aspect is, of course, to say, let's use behavior change techniques to modify people's relationship and behavior with respect to light. Because these behaviors, I think, can be modified, and we've been working on behavior change technology using apps in which we're combining sensing with easily implementable light exposure recommendations. Now, I'll just kind of position these two different approaches against each other and then see how we can move forward, right? So we can measure biological capacity by parametric changes in light exposure using controlled conditions and behavior, which generates mechanistic insight. On the other hand, the real-world relevance is, of course, a different question, right? So light exposure in the real world and its effects is messy and noisy, many things that can influence us in our circadian system. And so behavior is really important, I think, in modifying this. And so in my view, I think the way that we can bring these together is, of course, by doing both, right? I don't think we should focus only on mechanistic studies or going straight to implementation. I think we need to have, of course, an understanding that bridges the two. And one of the ways to do this is, of course, through recommendations, guidelines, practice, and communication. I'll highlight a couple of things that we have done in this domain that help us with this. But of course, what connects these two is we need physiologically relevant measurements, right? We need ways to capture light exposure in the real world that is with respect to the photoreceptors involved. With Ray and another group of people across the world, we've developed, and this is kind of a side note, a reporting guideline for documenting light exposures in the lab. This is currently under review, and we're just finalizing the revisions. But basically, these are the first guidelines that we can now use in the laboratory to document light conditions in the lab. It's based on a consensus, a statement. I just want to highlight a couple of communication areas that we've worked on. One of the things that we've done is a free comic book on the biological clock called Enlighten Your Clock. You can download it in English and I think 23 other other languages for free. If you do like it, please do get in touch. We're always happy to get good and also negative feedback, right? So keen to hear from you. And of course, we're trying really to bring the knowledge that we generate in a lab in the field to the real world. In closing, I want to highlight a project that we've just got funded with Ray. This is looking at optimizing light exposure in a scalable manner to do two things. We want to look at preventing myopia and we want to promote healthy aging. And so this project has just kicked off and we're hiring, and I think Ray has some information as well if you're interested to get involved just after the talk. So in closing, I just want to thank all the people that made this work possible. This is my current team at the Technical University of Munich and in Tübingen at the Max Planck. This is my dog. She's currently with me in my office sleeping. I don't think she's sleeping because it's the content wasn't interesting. I think it's just a natural sleep time. But yeah, so my team is very important. We've pushed, kicked off about a year and a half ago, and we got many different projects that are running, yielding already very interesting results. And with that, I want to thank you and in fact come back to the questions that we asked at the beginning. So let's see the percentage and the results of what we have. So can we see the results here?
So Manuel, I don't know if you can see the results, but I can. So for the first question, the photopigment rendering the intrinsically photosensitive retinal ganglion cells, only 27% answered melanopsin, which is the correct answer. Then the second question, what does illuminance correlate with? And this is perception of brightness, and here we have 60% who answered correctly. 26 who said circadian effect of light, and then color perception, 14%. And the last question, what is the influence of evening light on melatonin, which is, we have it popping up a lot in the questions now, is luckily only 8% said no influence. And then you have 56% who answered correctly, saying the suppression of melatonin production by light. Okay, but we do have 36% who said facilitation of melatonin production. So I think this is an important thing to address, right? Light at night suppresses your melatonin secretion, and melatonin is involved in so many functions in the body. It's also an antioxidant. It helps you, let's say, promote sleep. But basically, I think I will. Manuel, do you have anything to add before I conclude and we move on to the Q&A?
No, I mean, I think it's just interesting. I think from us who have worked in this topic, right, of course, is melanopsin, but I've definitely also seen, you know, a lot of people who, I guess, overall, I think to turn this into a constructive way of speaking about this, I think there's a clear need, I think, for us trained in vision and circadian rhythms to communicate our results in such a way that this knowledge that we have, that we intimately work with, is also translated to practice, for example, right? So this idea that we have different photoreceptors apart from the cones and rods, right? I think this needs to become textbook knowledge in optometry, in ophthalmology. And so I think there's a lot for us, I think, to do good communication, good science communication. But yeah, I think this is to me, you know, more an appeal to develop better communication strategies and tools. So happy to move to the Q&A, and thank you for listening.
Excellent. Thanks a lot for a really insightful talk, Manuel, as usual. I would like to invite my colleague from the Department of Ophthalmology, Assistant Professor Daniel Day, to join me in this panel discussion. We have around 15 minutes, but we have a lot of questions. So I'm just going to ask a very brief, maybe a very brief answer, maybe yes and no, Manuel. Is the current are the current indoor lighting options that we have outdated for when it comes to the circadian system? Are we just dealing with lights that are just made for good vision or good visual performance, but not at all for synchronizing your clock and preventing, helping your sleep, for example? Is that what we are dealing with at the moment?
Absolutely. I mean, I think there's a big revolution that kind of needs to happen in the way that we appraise and evaluate light sources, right? So we do have quantitative tools available, but when I, as a consumer, go and buy a light bulb, I get things like lumen, and I get things like CRI, or correlated color temperature, all of which is with respect to the visual system. And I think there's a lot more that we need to do to be able to say, okay, is this good in terms of the melanopic effects? And so I think this knowledge, I think, in the lighting industry is, of course, there, but without any regulation or without any guidelines, of course, it will not be turned into metrics that are actionable, right? And so I think there's a lot more that needs to be done, and a lot more that can be done.
Excellent. So before I move on to Daniel, you may have a question. Maybe can I ask you, Manuel, to stop sharing the screen this way? We can, yeah. Okay, excellent. So Daniel, do you have any questions?
Yes, thank you, Manuel and Raymond. I think this is a wonderful talk and a little bit different from our usual Envision talk, where we are focusing a lot on the health aspect of the light. We have numerous questions here, but I'm going to start off with questions that are related to your studies. So first of all, are these studies worldwide, and are they limited to a specific race? European, Caucasian, or are your subjects also inclusive of Asians and other races? Yeah.
So I think this is a really, really great question. So I think and I think it highlights a gap in how these studies have typically been done. The recommendations from Brown that we developed was largely done in either Western Europe or in the US. And of course, that basically then replicates the composition of ethnicity and race there. But I do think there is a huge study that I think one would need to do, which is, how does generally diversity factor in these responses? And I think we have similar, I guess, a similar skewed evidence base for many other different functions related to vision as well. But specifically, I think there's a need to run global coordinated studies under similar protocols to make an evidence base that's representative of different diverse populations.
Okay. I will just move on to one more question also from the audience. I'll combine two questions. So, and this is about light at night. So what happens to the melanopsin system in people? Basically, can we use this light exposure knowledge as a tool to aid patients with insomnia? And then the other question may be linked as well. We have three common light colors or correlated color temperatures, right? 3,000, 4,500, 6,000, probably sold on the market at the moment. How does one use this as a way to facilitate evening comfort and sleep? Goes back, combined, Manuel, with the insomnia question, please.
Yeah, I mean, I think this is a really good point. So I think with insomnia, of course, there's certain, it's not an easy mapping of saying fix your light exposure and your insomnia will be fixed, right? Insomnia can have different causes, but, and of course, there is comorbidity with many, many other diseases that can cause insomnia. So I certainly wouldn't say fix your light exposure and your insomnia will be cured. I think that would be a very naive conception of insomnia as a disorder. However, I do think that light exposure in the evening is, of course, correlated with a couple of things that might be conducive to insomnia. If we're using our phones, for example, we're using them because there's a behavioral component to it, right? We want to engage with the phone. We're hooked in some way. And I think it's a psychological component that might also then play a role. Of course, light hygiene or, having the right light exposure at the right time can overall support health and well-being. I think for looking at specifically disorders, I think one would have to be very, very clear on what is the evidence base. Of course, light therapy has been used for cure of seasonal depression. for a while. But I think, again, for insomnia, I think there is probably much limited evidence base. In terms of light source colors or correlated color temperatures, this is a very, very good question, right? So we can go to the shop and buy 3000 Kelvin lights that are orangish, or we can get daylight lights. The exact question, or the precise question, the precise answer to this question is, it depends on the spectrum, right? So exactly knowing what the spectrum is going to help us understand what is the melanopic effect. But generally, when we're looking at, for example, only LEDs, of course, with lower correlated color temperatures, we have a lower effect on melanopsin. That's because to make it orange, to make it 3000 Kelvin, you need to deplete it of short wavelength light. And so it's almost by accident, not by design, that these lights will produce less melanopsin excitation. And so in general, you know, I think the guidance would be to reduce your activation of the melanopsin system, use warmer lights, because most of the LEDs will produce low melanopic lights at these low color temperatures.
So can I add maybe to that point as well, Manuel? But you would advise in general to avoid getting any stimulation at night. So even if you're under, you know, low correlated color temperature light or orange light, let's say, and you're using your phone or watching a movie or getting some, you know, cognitive activation done, this will not help you sleep even. So don't necessarily count on the light if you're doing something stimulating, right?
Exactly. I mean, if you're watching a horror film just before going to bed and you do it in a dimmed way, I don't think it's going to help you, right? I mean, I think there's some balance, and I don't know if we know that function between the brute light stimulus and then whatever psychological content that might activate you.
Excellent. Can I ask a question by my colleague here at SNEC, Dr. Rachel Chong? Is there a difference between exposure to devices emitting high light levels in the dark versus ambient levels of light, ambient high levels of light?
So this is a very interesting metrological question, because if you had a phone close to your face and it was bright, or you just looked at ambient illumination of a wall, you might get the same lux values. And that's just because, you know, the way that the lux unit works, or the melanopic EDI works, is that it integrates across space in such a way that it kind of washes out any differences in what the scene is that you're looking at. You might well get differences physiologically in your responses to these two stimuli, but I don't think we have a full understanding yet of how stimuli of different size will influence the melatonin, or will influence the melanopsin system, and in turn melatonin suppression. And there have been some studies that looked at very bright light exposure from these displays, finding a negative effect. But exactly, you know, how bright this has to be with respect to the size of the display, I don't think we have a good model or prediction system for. So I think I would expect differences, just because, you know, of course, the stimulus to the retina is going to be different, whether you have something that's expanded or something that's small but brighter. And, you know, I think there's more work that I think would need to be done on examining exactly how these spatial factors contribute.
Thanks, Manuel. Daniel, do you have any questions?
I think there's also another question from the audience. Will you be conducting studies on people working 24 to 36 hours shift, like our medical officers or flight stewardess? What kind of
Sorry, can you repeat that? I had my connection dropped here for a second. Can you repeat the question?
Oh, yeah. Have you studied, or will you be conducting studies on people who are working on long shifts, like medical officers and flight stewardess on 24 to 36 hours shift?
So currently we're not running any of these studies. We're basically focusing on people who generally work during daytime hours, but I think there's a big need, I think, for systematic studies looking at people who work long shifts and who also do shift work, right? I think there's a big question of how can we support light exposure and also circadian and sleep health in general. I think this requires some dedicated attention. Currently, we're not running any studies in the lab in this domain, but certainly you would be quite interested in developing this branch as well.
Excellent. I think this is a tricky question, but we have to ask it. What are the current recommendations for setting of brightness throughout the day when using a light-emitting device? This is tricky.
Yeah, this is very difficult, right? I mean, I wish I had the solution. I wish I had the algorithm. I think, ultimately it really depends on what kind of model you might pursue. My feeling is that you could start with the Brown et al. Recommendations, where you say you need at least 250 lux melanopic EDI during the day, and then pre-sleep environment, you have less than 10 lux. Of course, this depends very much on what lights there are, what people are doing in the space. And so the problem is a little bit unanswerable unless it's with respect to a specific group of people. Added to this, of course, different people with different chronotypes will need different solutions, right? So if I have someone who's a very late chronotype and I start ramping up their bedroom at 7 a.m., which might work for an early chronotype, of course, this is going to be counterproductive. And so ultimately, I think recommendations we can use to build systems, but I think there's an empirical question of, under different scenarios, which types of light levels should be used for specific groups of people.
Got it. And maybe concurrently to this, and this is a topic that's been brought up many times, would blue blocking glasses help out in any way during the day or during the evening?
This depends very much on what type of light is filtered, right? It depends very much on the transmittance. And I think that it's a little bit of a problem. I think that the term blue blocking isn't technically defined, right? So I have colleagues who think of blue blocking glasses as the amber orange glasses that some people might put on before going to bed. But of course, there's also the ophthalmic glasses that people, or the blue light filters that are now in most prescription glasses. And these will have very different effects on the light stimulus. So the answer depends very much on the transmittance of the filters, right? And I think so. Just recently, there was a Cochrane review looking at the effects of blue light filters on a bunch of different visual and non-visual tasks. I think concluding that we need more research, and I think this is something that, of course, I would underwrite. I think it depends exactly on the light exposure you get, the transmittances you get, and also the prior light exposure you would have gotten. In any case, I think light during the day, barring UV exposure, right? You generally want to have bright light during the day, because that helps you become a little bit more resilient in the evening and also makes you more alert. Of course, barring UV and sunburn risk and many other factors that might be related, for example, daylight. But yeah, I think that would be my answer is, unfortunately, it depends exactly on the conditions and the transmittance functions.
Maybe one last question or a couple more. Maybe we have 2 minutes. Depends on how fast the answer is. What happens to the circadian system for people who live in the far north with white nights, or actually sometimes full days of sunlight? So maybe people living in the Antarctic, in the Arctic, closer to these regions. What happens to their circadian system? Elena's asking.
This is a really good question. I mean, I think I would say I'm not an expert on this, but Ray, if you have something to say to this, I mean, I would expect there's long-term adaptation processes in the melanopsin system and also long-term effects on circadian and sleep that probably recover by the time that half a year has passed. But I think, if you have what to say,
Yeah, definitely. So there are studies that have been conducted, and it's quite difficult to talk on the long term. I agree with you, Manuel. But on the short term, for example, scientists who live in the Antarctic, they started noticing that some of these scientists started sleeping at a very weird timing. So they started drifting, sleeping later and later every day, waking up later and later every day. So their circadian systems was not entrained or was not synchronized to light-dark cycle, because it was nighttime all day long. And these were studies, again, done in the Antarctic. And then we actually conducted this study where we boosted the artificial light with blue enriched white light and showed that we can actually manage to maintain some entrainment, some synchronization of the sleep and wake time, some synchronization of the melatonin secretion profile in these scientists in the Antarctic. So light plays a very important role for the circadian system, and to depriving people from proper light-dark cycles and proper intensities of light is a problem in the Antarctic and the Arctic, or even countries who are closer to the north and south. So yeah, okay. I think we have a lot of questions, probably the most question in this ever. Let me see, maybe last one. Question from Swapnil was whether the effects of monochromatic blue light and blue enriched white light will have similar effects on circadian rhythm.
I think this depends very much on the melanopic EDI, right? So I think you can find an equivalence in which a monochromatic blue light and a blue enriched white light will have the same melanopic EDI. And if we believe these to be, if we believe melanopsin to be the primary driver of this, then of course, it all depends on the intensity. So I think in a melanopsin weighted way, I think it should have a similar effect.
Got it. Daniel, do you have any more questions, or shall we conclude? I think we still have a lot of questions from the audience, so I apologize if we're not answering all your questions. We did answer as many as we can. I answered some of them by typing, Daniel, as well, and Manuel answered quite a lot. This was a very exciting session. Daniel, anything from your side?
Probably I have one question, because melatonin is involved in a lot of the health aspect, including if maybe perhaps it's not mentioned yet, blood pressure control and mood as well. So do you foresee, what do you think is the reason that the melatonin drops across age? Melatonin level drops across age?
That's a very good question. I mean, I think you do see calcification of the pineal gland across age, but I think to find causal relationships, I think is really tough, right? I think one would have to do epidemiological acrobatics, I think, to be able to find a causal relationship. I think in animal models, I'm not so sure, but it's clear that these are things I think one would need to consider across age specifically. But I don't have a definitive answer, unfortunately.
Okay, thank you.
Okay, I guess with that, Manuel, we thank you so much for this amazing talk. Very nice interaction, very nice questions from the audience. Thanks everyone for attending. I think we have a couple of announcements before we conclude. And Daniel, thanks a lot for co-hosting this event as well. So for our next webinar, it will be held on the 10th of October, same timing, and it will be delivered by Associate Professor Carol Chung, and the topic is using deep learning for detection of Alzheimer's disease based on retinal images, and it will be hosted by Assistant Professor Tammy Chung from the Department of Ophthalmology. Next slide, maybe, for a couple more announcements. Shalini, please. So, as Manuel mentioned, we just started a study in Singapore about improving light exposure in children, but also in children to prevent myopia. And this is particularly in classrooms. We're trying to improve lighting in classrooms to see whether we can stop or at least delay the onset of myopia in primary schools. And another project, Manuel will be focusing on improving light exposure in older individuals to build resilience against cognitive decline through healthy light exposure. So for both projects, actually these are the same project, it's called Lightspan, but for both studies, we're looking for manpower. We actually are looking for 3 research assistants, actually 6 in total. So if you are interested in myopia prevention in school-based studies, please reach out to me. If you're interested in light exposure in elderly and improving resilience against cognitive decline in elderly, please reach out to Manuel. We also have open positions for clinical research fellow and research fellow. Feel free to take a snapshot of that page on a print screen, and don't hesitate to reach out if you're interested in these job positions. And we have a next slide maybe, or that's it from our side. Okay, so with that, I would like to conclude for this webinar session. Manuel, thanks again for this amazing talk and for sharing all these exciting studies with us. And thanks a lot to our participants in our session. And Daniel, thanks a lot to you. And Shalini, as always, excellent job. Thank you so much. So with that, we'll see you in the next webinar.